Hello everybody, welcome to Bio 10, Week 2, Lecture 2. My name is Amrita Sharma and we will be talking about biomolecules today. So in the previous lecture we had an overview of what are the inorganic molecules of life, which is water, acids, bases. There are some salts but we didn't really talk about them. Now we'll talk about the organic molecules or the biomolecules of life, which are carbohydrates, lipids, proteins, and nucleic acid. And before that, we have to talk about carbon, the very, very important atom of life. So carbon forms the basis of almost all biomolecules. It has a very unique property of making carbon-carbon bonds. Now carbon has 4 electrons in its outermost shell, and here you can see the circle ones are the electrons of carbon and the crosses are the electrons of hydrogen. So it can form 4 covalent bonds. Here it's forming 4 covalent bonds with 4 atoms of hydrogen, and it's making C1H4, which is methane. Carbon can also make really long chains with itself and with other atoms, which is why it can make macromolecules, the huge molecules of biological system. There are several functional groups. The functional groups are specific group of atoms which provide different chemical properties to a molecule. For example, here you have R would just mean a long chain of C and H. The functional groups are these. Here hydroxyl functional group is OH, carboxyl is COOH, amino group is NH2, sulfhydryl is sulfur and hydrogen, and phosphate is this phosphorus with 4 oxygens and either some negative charge on them or hydrogen bound with them. Building blocks of biomolecules are called monomers, and when monomers come together and join, they form polymers. For example, polymer is protein and its monomer or the building block is amino acid. Building block of carbohydrates is monosaccharides. Building block of lipids is triglycerides, which is fatty acid and glycerol. And the building block of nucleic acid are nucleotides. So how do these monomers form polymers? So here is a glucose molecule. It's a monomer. This is a fructose molecule. This is also a monomer. They will lose one molecule of water here in a dehydration reaction, and they will join and form a molecule of sucrose. A molecule of sucrose is then undergoing hydrolysis by addition of this water molecule, which breaks this bond and puts OH on one side and H on one side and separates out glucose and fructose. So first category of biomolecules that we are going to study today are carbohydrates. Carbohydrates have carbon, hydrogen, and oxygen, typically in the ratio of 1:2:1. So here for 6 carbons you have 12 atoms of hydrogen and 6 atoms of oxygen in all these 3 molecules of sugar or carbohydrates. So this is glucose, this is galactose, and this one is fructose. If you have only one molecule of sugar, it is called monosaccharide, which is simple sugar. If you have 2, then disaccharide. If you have many, then polysaccharides. Carbohydrates have several functions in our body. The most important one is providing a quick source of energy. So, what are the types of carbohydrates? First is the monosaccharide, in which you have only one molecule of sugar. They are called simple sugar, and their general formula is one atom of carbon, two atoms of hydrogen, and one atom of oxygen in multiples. These sugars are polar. You must have seen that the glucose you have in your kitchen can dissolve in water. Common examples are glucose, galactose, and fructose. Disaccharides are double sugars where you have two monosaccharides coming together forming a bond. For example, glucose and fructose makes sucrose. Glucose and galactose makes lactose, and two molecules of glucose makes maltose. Sucrose is the sugar that you use in your kitchen. Lactose is the sugar of milk, and maltose is generally used for brewery to make beer. And here you can see how glucose and fructose remove one water molecule and fuse together to form sucrose. Polysaccharides are many molecules of sugar coming together to make long chains. These chains can be branched or unbranched. Examples are starch, which is the form in which glucose is stored in plants. Glycogen, glucose is stored in animals in the form of glycogen, because you can put together many subunits of glucose in the form of a biomolecule, a huge chained complex structure. Cellulose is the polysaccharide which is present in the plant cell wall, and chitin is the polysaccharide which is present in the exoskeleton of insects. Like you would have seen those plates or shells on top of some insects, the hard structure, they are formed of chitin. What are lipids? Lipids are oils or wax or fat. They are hydrophobic. They do not dissolve in water, as you have seen in your kitchen also. Butter and oil do not dissolve in water. Things that do not dissolve in water are generally non-polar. Because they are non-polar, they don't have charges on them, which is why they are hydrophobic. They sometimes may have some hydrophilic functional groups, but they are very few, which is why they mainly remain undissolved in water. Common examples are fat, which are solid at room temperature, and oils, which are liquid at room temperature. Fat, you have seen there is a lot of fat stored in animal body under your skin, and oil is what is produced around your nose or in your hair or in the feather of animals, which prevent water from setting on their wings, because if a lot of water sits on their wings, then they will have hard time to fly. The monomeric subunit of lipids is called triglyceride. Triglyceride is one molecule of lipid. The triglycerides are made from one molecule of glycerol and three molecules of fatty acids. So they come together to form one molecule of triglyceride. So these triglycerides can be saturated when all carbon have hydrogen bonds everywhere and they do not have any double bond, and unsaturated if they have one or two double bonds and they don't have hydrogen filling up all the bonds in with carbon. So the saturated fats they do not have any double bonds. For example, this here you see that there are all single bonds, single bonds, single bonds in your saturated fats, and unsaturated fats which may have a double bond. Like for example, here you have a carbon-carbon double bond. So because they do not have, these guys do not have double bond in them, they can make very tight packing with other molecules, which is why they are solid at room temperature. Whereas these guys have kinks, they have hard time packing with other molecules, they are liquid at room temperature. So the saturated fats, which are usually solids at room temperature, they tend to stick to the blood vessels and cause arteriosclerosis, high blood pressure, and even heart attack. Whereas the unsaturated fats, which are liquid at room temperature, are considered healthier because they do not stick to your blood vessels. So now this might seem a lot of chemistry, lot of information, but it will be easier as we go on. Next category of lipids are phospholipids. Here you know that we had one molecule of glycerol and 3 molecules of fatty acids in a triglyceride. When you remove this one molecule of fatty acid and instead put a phosphate, you get a phospholipid. So this is a lipid molecule with a phosphate. So this side of molecule which is charged is hydrophilic. This side of molecule which is non-polar is hydrophobic. Here is how these phospholipids are arranged in the plasma membrane. The charged phosphate site remains outside because it's hydrophilic, and this side, which is the long chain of fatty acids and is hydrophobic, remains inwards. Lecithin is another kind of lipid which is found in cell membrane, and it's used as an emulsifier in food production. Steroids is the next category of lipids. Steroids have characteristic 4-ring structure. You have 1, 2, 3, and 4 rings. Steroids are cholesterols. A common example of steroids is cholesterol. Cholesterol is, you all must have heard about cholesterol many times. Cholesterol is used to synthesize steroid hormones, bile, and vitamin D3 in your body. So this is cholesterol. The steroid hormones in your body are sex hormones, which can be progesterone, testosterone, and estrogen. They can be corticosteroid hormones, which are secreted by the adrenal cortex of the adrenal gland. And you have androgens, which are the precursors of sex hormones. Men get their secondary characters, they have masculine characters because of the presence of androgens in their bodies. So these are the cholesterols, which are the steroid hormones. So this is the cortisol, this is the testosterone, and this is the estradiol, which are basically the sex hormones, and their basic structure is a steroid structure. A steroid structure is 1, 2, 3, 4 rings of carbon. Next biomolecule is protein. Protein is made up of amino acids. There are 20 different kinds of amino acids which make long chains with the bond called the peptide bond, and they make proteins. If there are only 2 amino acids, it make a dipeptide, 3 amino acids make tripeptides, and many amino acids come together and form a polypeptide. What is the structure of proteins? A straight long chain of amino acid is the primary structure. They may form a coil like this or a zigzag sheet like this. These are the secondary structure, the alpha helix or the beta pleated sheets. These structures can then form the tertiary structure, in which this secondary structure further coils. This tertiary structure then come in contact with other proteins, make a three-dimensional structure with multiple protein chains, and they form the quaternary structure. Sometimes because of heat or change in pH, this 3D structure of protein get destroyed, and this causes the denaturation of protein. Proteins lose their biological function when they lose their specific structure because of heat or pH, which is why it is very important that your body maintains a pH and a specific temperature. Proteins can also conjugate with different sugars, make glycoproteins, or different lipids and make lipoproteins. So what are amino acids? Amino acids are, here you have carbon. Carbon is bond with more carbon chain on one side and NH2 side chain on one side, which is called amino group, and COO negative on one side, which is the hydroxyl group, which is why it is called amino acid. These amino acids make connection with other amino acids through a dehydration reaction, and they form long chain of peptides. So amino acids, when they form covalent bonds with removal of one water molecule, they make polypeptide chains, and these polypeptide chains then fold and become proteins. Now about the functions of proteins. So proteins display various functions in our bodies. These are structural proteins such as actin, myosin, troponin, which are present in your muscle fibers. They have enzymatic role. For example, they help in digestion. Trypsin, pepsin are involved in digestion. There is DNA polymerase, which is involved in synthesis of DNA strands during DNA replication. There are hormones which are proteinaceous in nature. Then you have signaling molecules which gives a signal to the cell for doing a specific function. For example, you want the cells to start dividing, start multiplying. You get a signal from outside. These can be like growth hormones. For example, growth hormone gives a signal to the body that now you have to grow, become bigger. So cells start to divide when they receive a signal from a signaling molecule. Then you have antibodies. Immunoglobulins are the antibodies produced by your immune cells, the lymphocytes, the white blood cells in your blood, and they defend your body from foreign antigens. They help in maintaining fluid balance in your blood. They also help in maintaining the acid-base pH in your blood. They are also present on the membrane of your cell, and they act as channels or pumps or transporters in which they let only certain kind of material to enter your cell, because entry of every other kind of material inside your cell would be detrimental. Fourth and last kind of biomolecule that we will be studying in this class is nucleic acid. So nucleic acid are the macromolecules which carry the genetic information. So the genes which are passed from parents to the offsprings, the characters that pass from parents to offsprings are basically the basically the nucleic acid is responsible for that. So nucleic acid are of two kinds. They are DNA and RNA. The basic subunit of nucleic acid is called nucleotide. So this is how a nucleotide looks like. So nucleotide will be one molecule of sugar that you have seen before. You have one phosphate molecule on one side and nitrogen bases. What are these nitrogen bases? These nitrogen bases can be purines or pyrimidines. Purine are two ring structures like adenine or guanine, and pyrimidines are one ring structure, which are cytosine, uracil, or thymine. Let's first talk about DNA, the first type of nucleic acid. DNA is the genetic blueprint of all living organisms. They are passed from parents to offspring, and they carry the genetic information, what kind of proteins to synthesize, what kind of characters to carry on. DNA is made up of nucleotides, and these nucleotides are made up of deoxyribose sugar, this one, this thing, and four kind of nitrogen bases. Adenine, which is this guy, the double structure, or guanine, double ring structure here, or cytosine, the single ring structure here, or thymine, the single ring structure here. Now guanine makes a triple bond with cytosine, and thymine makes a double bond with adenine. The whole sequence of these bases gives the information to our body to what kind of genes to express, what kind of proteins to make. And the human genome contains around 3 billion of such pairs. So this is one pair, this is two pair. There are 3 billion such pairs in the human genome. Let's look at the structure of DNA. So here you have sugar molecules, and in between there are phosphate molecules. So this is a sugar phosphate backbone. So this is a sugar phosphate backbone. Then you may have adenine, cytosine, thymine, or guanine. The antiparallel strand, the strand opposite, also has sugar phosphate backbone and can have thymine where it's adenine, guanine opposite to cytosine, adenine opposite to thymine, and cytosine opposite to guanine. So this double ring structure pairs with single ring structure. Every time a double ring structure would pair with a single ring structure because they want to keep the distance between these two strands equal. Right? Then you see that carbon has this numbering 1, 2, 3, 4, 5. 3, 5. 3, 5. That's why this chain is running from, this is 3 side and this is 5 side. And here you have 3 here, 5 here, 3 here, 5 here, 3 here, 5 here. So 3 to 5 is this direction. But DNA strands are always read in 5' to 3' direction. So just remember that. Now, if we just make a summarized version of this thing, you have two strands which are sugar and phosphate backbones, and let's consider that this is a ladder, and the steps of these ladders will be equal length steps made by the bonding of one purine with another pyrimidine. Here you are looking at the coiled structure. Now this ladder will further coil up and form the actual packing of DNA, this structure coils up because a very, very long strand of DNA has to coil up in a very small space because it contains a lot of information. Think about it. The entire being you are, how tall you are, what is your skin color, what is your hair color, what is your eye color, all the diseases, the genetically inherited diseases, all that information is here in this DNA. So such a long DNA has to be packed up in a very small space. That's why it is coiled. The other kind of nucleic acid is RNA. RNA is mainly required for taking the information from DNA, and that DNA information is used for making proteins via RNA. We will probably learn more about this in subsequent lectures. So the DNA stays inside the nucleus of your cell. It only makes a copy of RNA that goes outside and makes proteins in the cytoplasm. RNA has some differences from DNA. One, RNA is a single strand structure and DNA is a double strand structure. The sugar backbone is different. DNA has deoxyribose sugar and RNA has ribose sugar. As you can see, there is one oxygen less in DNA than in RNA. So it's a deoxy form of ribose. The other difference is instead of thymine of DNA, you have uracil in RNA. RNA is in turn of three kinds. One is messenger RNA, tRNA, and rRNA. Messenger RNA is the one that takes the message from DNA in the nucleus, brings it to the cytoplasm, and this is the template on which proteins are synthesized. tRNA carries specific amino acid, bring them to the ribosomes which are sticking on this mRNA. As the ribosomes keep reading this mRNA, this tRNA keeps bringing newer amino acids to make a protein or a polypeptide chain. And this ribosome is made up of ribosomal RNA and some ribosomal proteins. So ribosomal RNA is integrating with the ribosomal proteins to make larger and smaller subunit of ribosomes. So all these three kinds of ribosomes have very essential function and different functions in the process of protein synthesis. Collectively, they perform the function of protein synthesis. One last point is a very important form of a nucleotide. This nucleotide, which is sugar, nitrogen base, and phosphate, is called ATP. And ATP, as you know, is the energy carrier in our cells, in our body, and it's synthesized during the process of cellular respiration. So here your lecture ends, and thank you for tuning in.